I'm with USDA in Manhattan, Kansas. I've been there about 20 years. And since we're at the Borlaug Global Rust Initiative, in 2004 I was at Summit and we were there for a Carnal Bunt meeting. And one morning we had breakfast and we were all sitting there and we looked over and somebody said, "I think that's Dr. Borlaug." And I said, "Come on." So we went over there and we introduced ourselves. He said, "Please sit down." And we all went around, told about ourselves. And I said, "Dr. Borlaug," I was like, "I'm a molecular biologist." And I said, "You know, I'm not your typical breeder." And I was like, "When you see in biotechnology and things like this, what would you suggest for somebody like me that's working in wheat diseases?" He said, "Try to figure out why rice can't get rust." And so that's always been in my mind and what I do. And so unfortunately, somebody else took that project. So I'm trying to do something very similar in wheat. Looking at the susceptibility, and I have poster number 47 back there where we're looking at mutants looking at the susceptibility of wheat and how leaf rust lives inside of wheat. But today I want to talk about the work that I've been doing on effectors. And so since we're a mixed audience, I want to take just a little time and kind of get everybody on the same page. So what we have in pathogens, especially rust, is that each one, we have these effectors. And these effectors are molecules that the pathogen squirts into the cells, and it basically reprograms the cells. It shuts down defenses. It makes that cell its own quickie mart, you might say. But these effectors, they have a purpose. And some of these are transcription factors. Some of them are promoter RNA binding. Some of them are protein modification. But we all know them and a lot of plant breeders in that we provide these major genes that recognizes a specific effector, and that effector binds with that R gene, and it turns, in most of the major genes, turns on apoptosis, and we see it as a fleck or a cell necrosis. But what happens is we use these R genes. There we go. So we use these R genes though, and we have a set of differentials. So in leaf rust, we have the Thatcher isolines, and the first set is Lr1, Lr2A, 2C, and Lr3. And we rate these and giving a letter designation based on the virulence on the pathogen. So an M race would be high on 1, low on 2A, 2C, and high on 3. And then a T race would have overcome 2A and 2C. So that's how we type them. It's basically, until the genomics, that's world and molecular biology, that's basically most of the understanding that we had on the biology. So one of the things that we first tried to understand and clone effectors was when we didn't have a genome is we used ESTs. And so we began sequencing leaf rust ESTs, which are basically copy DNAs. And so this is work that we just published a couple years ago by a graduate student, Vanessa Segovia, where she identified a group of ESTs that, this is not working very well. Anyway, that matched a given profile. They were small, they had secreted peptide signals, and so the first two, PT2 or PT3, PT12, and PT27, were our first candidates. And so we used a transient assay system where on the left column we see Thatcher 26, which is the isoline. On the other side we see Thatcher. And what we did was we co-bombarded this leaf tissue with a plasmid that expressed GUS. The other one expressed our effector candidate. The idea was that if the plant recognizes the effector candidate, it will kill the cell. If you kill the cell, you don't get GUS expression. So our first candidate, PT27, followed that, where PT3 and PT12, if you count the blue spots, there's no significant difference between Lr26 and Thatcher, but in PT27, we saw almost no blue spots. So this looked like a great candidate for us. And like I say, we published this. But there again, we didn't have the gene cloned. Lr26 is on a RI translocation, so it's going to be a little bit more difficult to clone. But with the new mut-RenSeq, it may be possible. So we set out in 2009, an opportunity came up to sequence the leaf rust genome. And so we worked, myself, Gus Baccarin, Les Szabo, and Christina Cuomo headed up a project funded by USDA and NSF to fund the genome. Puccinia triticina is the largest of the 3 genomes of the rust. If you see what's highlighted in blue, Graminis is 88 megabytes in length. P. triticina is 135, and P. striiformis was 117 megabases. So these for pathogen or for fungi are fairly large. But what was most significant in leaf rust was that 45% of the genome was repetitive DNA. So there I was working in wheat, which is mostly repetitive DNA. Now I'm working on another organism full of repetitive DNA. I'm a glutton for punishment. But anyway, we figured out that that P. triticina had around 15,000 genes. Of those, 1,358 had secreted peptide signals. So those are the ones that we are considering are effectors, and also ones that we consider avirulence candidates. But the problem with assembly was it's a 454 assembly, and these were in the super contigs, as we see up on, or the contigs, there's 24,000 contigs, which are simply how the computer pieced together the DNA. And then using back libraries and phosmids, we were able to scaffold these into about 15,000 super contigs. But in these super contigs, where the gaps were, they put ends. So there are about 309,000 ends in the genome that we have now, the version 2 genome from the Broad Institute. That can pose a problem because there's parts of the genome there that we don't know anything about. And another problem that we have is it doesn't deal with the dikaryotic nature of the genome. So the 2 haplotypes, they may be split up in a couple of contexts, but most of them collapse on one another. So what happens is when you take Illumina data from the same race, race 1, and put it back onto the genome, we still get almost 300,000 SNPs. So when you're trying to do some sort of SNP analysis or some sort of mutation, knockouts, these are major things that you have to take into consideration. This genome was released. We also did the PST78 genome that went, Zhumin Chen and Scott Hulbert from Washington State, we all put that together and did a whole 3 rust genome comparison. So as we look at the 2 genomes, the first thing you would say, well, what about, are there orthologues? Well, yes, there are. So the PTG0152 highlighted in orange shows that it has an SP score, which is a secretion signal score calculated by a program based on the first about 30 amino acids. It shows that a closer number to 1 means that it's very likely is secreted. It has almost a 90% alignment to a gene in PTG, or PGT, I'm sorry, and which also is, has a high secretion. So this is probably, these 2 are probably orthologues. But what happens though, in the ones in yellow, is they have high secretion signals, but either there's no orthologue in Graminis, or there's very little amino acid homology. So there we are stuck. It's kind of its own beast. So I apologize for the small letters, but when we did the genome, it was at a time when sequencing costs were plummeting. So we first started this with a 454 type sequence. It was $1 million project. We did a 30x genome coverage. And Christina called me up one day and she said, hey, we still have some money left. What do you got in mind? And I was like, well, let's do some races. So I contacted Jim Comer and we started putting races together. And before the project ended, we had sequenced 121 different races that Jim had collected from around the world from different time points and originated on different species like Aegilops, Tauschayi, Durham, and what I'm showing here are the three major clades of North America. The one up in green recently showed up in the late 90s when Kansas State released the variety Jagger. And we called this the major Jagger shift. So everything suddenly became high on 17. So, but what is also unique, as has been talked before, is that in North America, we believe that this is asexual. So this also provides us an opportunity to study how these mutations are happening. And maybe by following this tree is that we can trace that mutation back to the origin. Down in the light purple is where probably the oldest races are that are in North America and have most of the variety. So now we have all of this data. We have the SNPs from all of these races. So that's one piece of the pie. So we started taking these SNPs and started drawing associations. And it was simply me going through, I'm not that computer savvy, so I had it in Excel, color-coded all the SNPs, and then started looking for color commonalities. And so what I'm showing here is that the beige bands show regions that are very close together that have SNPs that are associated with various leaf rust genes or overcoming various leaf rust genes. And so you can see some of these SNPs actually are associated with several changes and not just one particular leaf rust gene. So this was a database that I built. And so then about 6 months ago, working with Edward Ochenov and Fei He, we took all of this SNP data and tried to run GWAS on it. And so we would hope that you would have a segregating sexual population, but I wanted to see if we could draw simple associations just from the SNP data and you draw some sort of statistical conclusion on any of these SNPs and whether they're associated with an LR gene. So what the graph on your right is showing is that the number of times that, or the number of contigs, and there's 15,000 contigs, where they find a SNP associated, and in the first bar is with LR1 or LR14B. So the ones I'm really interested in are the really short bars, so LR16, 28, and 3BG. So now the LR16, I think when I looked at the data, there were actually 41 SNPs there. So there's still a lot of data there. I mean, I could go in and I could clone up all 41 genes and try to run them through the expression or transient assay system, but that's still a lot of number. So we still needed one more way of reducing it down. And also, if you look at this, when we looked at the first 57 races, in the North American lineage, there were 115 SNPs associated with a change from Lr2A to 2C, going from low variance to high variance. And then in the same thing in North American 3 lineage, there were 104 SNPs. But when you did the comparisons, none of these SNPs lined up. It's all, as you think about it, it's logical. If these lineages developed differently and if they became virulent on Lr2A, it was probably an independent event and they probably would not share the same SNPs but may share the same gene. So we needed another way to really look at this. So one of the things I did is I exposed spores to neutrons. I think somebody else earlier today talked about that at Kansas State University we have a nuclear reactor on campus. So we took spores over to them and they exposed it with fast neutrons. We inoculated these spores onto a specific Thatcher isoline. We isolated the single spores, and I've showed these, and I don't know if you can see them that well on your screens, but I've highlighted them with the red circles. So this is an example for a mutant pustule for Lr2C and one for Lr26. Throughout this, I've been able to isolate mutants that overcome Lr2A, 2B, 2C, 11, 16, 17, 26, and 52. So it's a very efficient method as long as we have a good, clean HR. Anything like LRB where you see a 2 to a 2 plus, it's going to be very difficult to try to isolate it in this manner. So I purified these mutants, carried them through several lines of selection, and then isolated DNA from them. So this is just kind of brings it in a little bit more. And panel A is the mutant that I just showed previously. I take a Q-tip, a cotton swab, dip it in Soltrol, tap it onto that, and then spread it onto an Lr2C plant, which is in the second panel. This is after about 14 days. You can see it's built up. We continue to do this until we get enough spores that I can isolate DNA. And I'm not sure if it's real clear, but the cool thing about this Lr2C mutant is this Lr2, 1, 2A, 2C, and 3A is the first set of the differential lines. When we sprayed this particular 2C9 mutant, it turned out that not only was it high on 2C, but it was also high on 2A and 2B. So we know that Lr2A, 2B, and 2C are all allelic, or they're on 2D. And so through all this, I've identified 6 mutants that are 2A virulent, or virulent on 2A, and 7 that are virulent on 2C. This is more, this shows you the complete set of the differential lines, and I've highlighted in red the lines that have high virulence. So what this is showing is that using BBBD, which is the most avirulent race that we have available, it's down at the bottom, the BBBD wild type. You can see that all across this, it's high except on 14A. BBBD is also high on Lr20 and Lr50. So by highlighting here, I show you my mutants for 2A, 2B, and 2C. I'm also showing my mutants, 2 of them from AVR Lr11, and then 3 for Lr26. So this seems to be a very good method for getting very clean mutants. So once we got the sequence from those mutants, then we started piecing the puzzle together. So the first thing that I did was I went through and just looked at the secreted peptides, and I found 11 secreted peptides where I could find consistent mutants within the gene, whether they were, and most of them were overlapping, whether they were deletions or they were snips. Then I overlapped, so that's the first line. So the second one, since it looks like it's the same gene, I then looked at Lr2A mutants and found the ones that overlapped with them. Then looking at the SNPs, I looked at the genomic DNA for North American 5 races and found those. Then with races in North American 1 and 2, those races actually separate out Lr2C only. So these have been very useful. So I found the genes that overlap there, and then the P races and the S races, the P and S is difference in virulence on Lr2A only. So I've narrowed it down to 2 genes, and I was hoping to have my initial transient assays before I got here. Just didn't work out, so these have been set off for sequencing, and I'm now working on those. So because we have such an issue with the genome, we've seen a couple papers just recently where they've used new technology, the PacBio, which provides longer reads, to try to pull out the, using phasing, try to pull out the haplotypes. So I've taken a similar approach with Lr2A. I contracted with Dovetail Genomics, where they use a high C assembly. High C is simply based that inside the nucleus, it's a 3D world, and those chromosomes are moving around, and they have proteins that bind onto them. So what this does is cross-link those proteins, and then it comes out, and then they clip it, and so it forms a long piece of DNA, and then they just in-sequence those. So those act like a long physical map. It's a new way of putting the chromosomes together. And what I'm showing here is that in the middle column on the comparative assembly, we see that initially it was about 2.6 was our longest scaffold. Now we're up to 15 megabases. So we're getting fairly long. Then also in my lab, I've been using the new Oxford nanopore system. I've gone through about 60 chips now with not only with Lr2A, but with wheat, tobacco, and also with viruses. It's very efficient. I even had my daughter the other day loading it up. But from this, I've done a 28 coverage, 28x coverage, and I've got it down to 392 contigs, no gaps. So this is an amazing technology, but it still isn't phasing out the haplotypes. So we've also done with Goose Baccarin, we just got a 90x coverage of PacBio. Our first assembly on it is 1,004 contigs, and it looks like we're starting to pull out some of the haplotypes as the genome size is a little bit bigger. We're working on using Falcon and the unzip to finish that contig. So what does this mean? Is that what this graph is showing, and I really wish this would, okay. So what this graph is showing is that mate positions are based on that high C where the protein's bound to the DNA and they end sequence. So every little dot on the little purple dot that you see is where one of these mate positions are. But the blue line up the middle is where the majority of them are. So this is where it's piecing together the genome. And yes, this was the high C data was put back onto the Broad Institute, but you can see the white lines in between the gaps. Let's see if I can get this to work. Oops, there we go. So these white lines here show major super contigs, and if you start to look here, you can look at that sequence and it's starting to show centromeric. There are 20 to 22 major groups right here. We're starting to get down to chromosome level assemblies. So it's a very powerful technique. I believe in talking with Les Zabel today, I believe the estimation for leaf rust during some flow cytometry was 18 to 21 chromosomes. So we're getting really close. So I think that for genomes, for leaf rust, and for any of the rust, I think we're going to be really close in the next couple of years. So this is my lab. Right now I'm very short. Donald Trump hasn't let us hire very many. This is 2 undergraduates and I during our Robigalia celebration. And by the way, it's coming up in the next couple of weeks. So Myron Bruce started the mutation work. Vanessa Segovia did the variance gene with Lr2A, and then Elizabeth and Kaylin are my undergraduates. Like I said, I'm working with Christina, Jim Colmer, Goose Baccarin, Les Zabel. David Cook is a new faculty member that came out of Art Thoma's lab. He's been helping me with the assemblies. And then Tom Swale at Dovetail Genomics. I'll take any questions at this time. Thank you.